Thank you for coming to my talk today. Today I would like to talk about quantifying soil microbial structures and functions for soil health assessments. First, I would like to thank our funding agent, USDA Natural Resources Conservation Service. This is part of a larger project called Dynamic Soil Property for Soil Health. It was initiated by NRCS a few years ago, and there are over dozens of other cooperator universities across the state. The main goal of this project is to assess the reliability and usefulness of common sets of soil health indicators measured across a wide range of soil and management conditions. So, what is soil health? Here's the definitions provided by NRCS. It speaks to the importance of managing soil as a living ecosystem so that it's sustainable for future generations. To do this, we need to remember that soil contains many living organisms, particularly soil microbes, that perform important functions. NRCS also published technical notes with a list of recommended soil health indicators. In this list, you can see that it contains many measurements that are related to soil biology and soil microbes in particular. So the question is, are these biological indicators sensitive enough to distinguish soil with different soil health practices? Also, DNA sequencing is a widely used method to provide detailed information on microbial diversity and community compositions. And we want to know whether the detailed information on microbial diversity will be valuable or useful for soil health assessment. In Connecticut, we sample soils from a gradient of management intensity, including a managed forest as reference habitat and managed forests that have been under regular thinning practices, grassy habitats as hay or residential lawn, cornfield under no-till practice for over 10 years, or cornfield was long-term conventional till practice. Within each management, we sample soils from 2 fields, and then within 2 fields, we deduct 2 to 3 patterns per field and sample 0 to 5, 5 to 10, and then generic horizons down to 1 meter. In the lab, we first quantify soil total carbon. You can see that from the top 0 to 5 centimeters, soil total carbon decline as management intensity increase. And the unmanaged forest has more total carbon than the managed forest. No-till soils has more carbon than the tilled soil, but hayfields have general similar total carbon as lawns. We also quantify the active carbon pool using the permanganate oxidation method. First, you can see based on the y-axis that POX carbons is a very small fraction of the total carbon, is only about 1%. And then we can also see that cornfields in general has lower active carbon than the other 2 land uses. We also quantify soil protein content using the citric acid extraction method. Then you can see soil protein content follows a very similar trend as the total carbon that has a very steep decline as management intensity increases. When we look at the 5 to 10 centimeter, soil carbon and protein content follow a similar declining pattern along the management gradients, but the differences among management systems become smaller as compared to the top 5 centimeter. And for POX carbon, there are no distinct differences among land uses. This graph is to show you that most of the soil carbon is concentrated in the top 30 centimeters. And you can see that there's barely very small amount of carbon below 40 centimeter, and there's no differences among different land use type as you go deeper down to the profile. And the next thing we look at is the microbial populations. We analyze the phospholipid fatty acid extracted from soil. Phospholipids are important components of the cell membranes, and different microbial groups differ in the chemical compositions of the PLFA markers, such as the number of unsaturated carbon bonds. Because PLFA decay within days after the cell die, the total PLFA is normally used as a proxy of the active microbial populations. And consistent with our predictions, we saw a decline of the PLFA as management intensity increases. And again, this effect is more apparent in the top 5 centimeters. We then further quantifying soil microbial respiration from short-term incubations of wetting the dry soil. We expect that the microbial respiration would be affected by both the carbon resources as well as the microbial population size. And consistent with these predictions, we saw a decline of the microbial respirations as management intensity increases. And again, the effect is most obvious in the top 5 centimeter. And when we look at the relationship among all these measurements, you can see that total soil carbon has the strongest correlation with all other indicators. The upper right panels of this graph show the Spearman rank correlation coefficients between pairwise measurements. You can see that soil total carbon in general has very high correlations with other measurements except for active carbon. The next thing we look at is the community structure of the microbial community. We first look at the fungal and bacterial ratio, quantified by the fungal PLFA markers and the bacterial PLFA markers. We hypothesize that this ratio is going to decrease as management increases. This is because fungal hyphae are more sensitive to soil disturbance than bacteria. However, in contradictory of predictions, we actually saw an increase in these ratios as management intensity increases. This is surprising, and when we look a little bit deeper at the results, we saw that the increase in this ratio is because bacterial PLFA decreases to a larger extent in response to management intensifications. How about bacterial diversity? We first look at the OT richness, the operational taxonomy units based on 97% similarity of the 16S sequence. And surprisingly, despite of the smaller population size in the cornfields, we actually see a higher number of OTU recovered from cornfield as compared to forest. And this results in a negative correlations between the bacterial population size using the PLFA method and the bacterial OTU richness. And we think that few factors may contribute to this inconsistency between the population size and OTU richness. Because for the bacterial diversity, in addition to the food supply, they might also be affected by the habitat and the disturbance levels and the metabolic stage. And also the sequencing methods also pick up probably substantial relic DNA that may not be metabolic active in the current populations. And when we look at the fungal communities in the 0 to 5 centimeter, we observe that the OTU richness of the fungal ITS sequence is higher in grassy habitat than forests or cornfields. But then in the 5 to 0 centimeter, the fungal ITS richness from the unmanaged forest seems to be the lowest among all the other habitats. When we look at the compositions at the phylums level, first we can see that bacterial compositions are broadly similar among the habitats, with the most dominant bacterial phyla Proteobacteria, Actinobacteria, and Acidobacteria. And each of those phylums contains many taxonomy as well as functional diverse species. And as this one phyla, the Nitrospirea, contains nitrified taxa that oxidize nitrite to nitrate, and they seem to be more common in hay and in lawn compared to other habitats. Similarly, we saw broad taxonomy, broad phylums level similarity of the fungal ITS sequence across different habitats. And one thing is that there's a lot more unknown in the ITS sequence compared to the bacterial 16S sequence, and we have about 20% of the sequence that were not assigned to any known phylum. When we look at the details of the OTU compositions, we saw that land use is the main factor that separate the bacterial communities. However, pH is the important covariance. As you can see, the size of different symbols represent different pH values, and forest sites are normally low in pH, and croplands are generally have higher pH. I also wanted to point out that the sites are recently converted from agricultural lands, such as this forest sites and this lawn sites. Their bacterial compositions are more similar to bacterial composition from cornfields, suggesting the strong legacy effect of bacterial communities from past land use. And the effects of the pH and land use The effect of the pH and land use is more clear when we look at the bacterial OTU richness. As you can see, there's a very strong positive correlations between the OTU richness and the pH of the soil. And the legacy effect also seems to appear from this relationship. To sum up, soil biological health indicators generally show similar results and decline as management intensity increases. And soil carbons has very strong correlations with other general microbial measurements, suggest the redundancy among soil health indicators. We also observe that the response of soil microbial community to management intensity is very complex, driven by currents and historical land use and the resulting change in the soil pH. Due to this complexity and difficulty in providing clear interpretations, we think DNA sequence at this stage might not be a good choice as soil health indicators. And we are still trying to understand all the results we have, particularly the soil microbial sequencing data. But what is clear and also have been demonstrated by a lot of other studies is that management practice and land use can change the food resources, habitat space, and other chemical characteristics for microbes that can affect the microbial population size and their community structure and compositions. But there's a lot unknown in terms of the exact response of microbial community compositions to management practice, and we need to identify whether there's any clear links between the microbial diversity and compositions to specific soil processes and functions. On the other hand, it might be more fruitful to develop measurements that can quickly assess specific soil functions, rather than trying to find reliable biological indicators that best represent change in their soil functions. Because such biological indicators may not exist or too noisy to capture. With this, I would like to thank all the NRCS soil scientists who have helped with the field work of this project. And this project was supported by USDA NRCS, specifically through this cooperative agreement awarded to the University of Connecticut. Thank you.